Thank you. Thank you for nice introduction, Dr. Mugini. First of all, I should say that this is a huge honor for me to receive this the honored award for the women in life science. Especially, I'm deeply honored to present my recent work. Actually, not my work; it's a teamwork, right? So our recent research on the thalamocortical circuit. So I named this talk to New Era for Oral Circuit because this sensory processing pathway from the periphery to the brain has been known for decades, actually, and during the centuries, right? And we studied especially the thalamus in the middle of the brain, which relay this sensory information to the cortical area. And recently we found that the interaction of the neuroglia interaction is also important to controlling this circuit. And we hope that this study will open up the new era for this old circuit. So before we jumping into the real data, I should introduce this brain circuit first because most of the audience here is not neuroscientists. So thalamus is located in the middle of the brain, and this is a huge structure to receive the sensory information, actually except the odorant, and relay that to the designated cortical area. And my lab has been studying this circuit for years and publishing the many papers on this sensory processing through the thalamus. And also this circuit is reflecting the consciousness level. Easily speaking, is vigilance, the conscious or unconscious state. Also, we studied the control of the consciousness through the thalamus for years. And today, I'm going to talk about our recent work on the neuroglial interaction in the thalamic area. And we found that the astrocytes in the thalamus control the thalamic sensory processing, and by providing the tonic GABA to the neurons. And also, I'm going to talk about what's the function of that. So we focus on the GABAergic signals, and GABA is the major inhibitory neurotransmitter in the brain, and it works in two kinetics. So first, phasic inhibition, which is very fast and very transient. And when you hear the GABAergic inhibition, maybe you can just think of this phasic inhibition, and mediated by the synaptic GABA-A receptors. And another mode of the GABAergic inhibition is exerted by this extrasynaptic GABA-A receptors, and it's expressed as a shift of the baseline. You can see that these green signals by shifting of the baseline. So this is a kind of the chronic and ambient kind of the inhibition. And I took this picture from the very famous review paper, and they are showing that this both phasic and the tonic inhibition is mediated by the GABA released from the presynaptic and terminals. So actually, this study started from the very simple question: is this always true? Based on this picture, we can easily think that both of the phasic and tonic inhibition will be mediated by the presynaptic GABA, so they should go parallel. But if I look up the literatures and found that the easily found the cases which is not the case. And I thought that if this picture is true ever, it should work on the thalamic the circuit, because thalamus has the prominent level of both tonic and phasic GABA. So in thalamus, this is where the biggest tonic GABA the signal is found, because the thalamus can be divided into the two nuclei, and this so-called TRN region here kind of embraces the other part of the thalamus, TC area. So the TRN is composed of exclusively with the GABAergic neurons, and they send a strong GABAergic input to the TC area. So people believe that all this huge tonic GABA current comes from the TRN neurons, so goes with the phasic inhibition together. So the presented with this stimulation of the TRN neurons increases tonic GABA current here. But from this study, we really begin to throw the question, is neurons, actually TRN neurons, the only source of the GABA in the thalamus? Because basal level of the tonic GABA is huge in the thalamus, but when they stimulated the TRN neurons with a vigorous electrical stimulation, they could increase the tonic GABA a little bit. So it really threw me the question, is this the real or not? So we started having this question. So we wanted to know the source of the GABA, especially the tonic GABA source in the thalamus, and what does the other glial cells doing to control this tonic GABA in the thalamus, and what's the function of that? So we started the experiment. We just incubated the brain slice with a synaptic vesicular release blocker for hours and hours, and repeated the recordings here. And this small flickering, looking traces are showing that the iPSCs, and this shifting of the baseline shows that the tonic GABA. And after incubation with the concanaminase A, which is the vesicular release blocker, this small flickering part is completely gone. So the basic, the GABA signals were gone, but tonic GABA signals, although it seems to be a little bit decreased, but it was almost intact, telling us that the tonic GABA is regulated in different way with the phasic GABA. So we look into the where it comes and the source, and looking at the EM imagings from the thalamic structure, labeling the GABA here, and we found that the astrocyte processes contains a lot of the GABA. Of course, it's a lot less than the GABAergic terminals, which are concentrated with the GABA. But this was we look up the researchers and found that this is the highest level of the GABA in other than the GABAergic terminals. But still, it doesn't tell us that this is releasable GABA or not. So we look into the feasible candidates for the release of this kind of the glial transmitter from the astrocytes. The first, the candidate vesicular release was out of picture from our previous data, showing that the concanaminase A doesn't block the tonic GABA. And transporter release was also, we did exclude the candidates by doing the experiment. And then we look into this second candidate, channel-mediated release, and found that when we measure the tonic GABA from the best one knockout, which are the calcium-activated chloride channel knockout mice, the tonic GABA level was significantly smaller here, and while the other phasic was not changed at all. And this channel was highly expressed in the astrocytes, and when we knock down this molecule in the astrocytic specific manner, it decreased the tonic GABA dramatically too. So from this set of the experiment, we could conclude that the thalamic astrocytes somehow that having the GABA and release it through the bestrophin channel. And also that they take up from the different set of the experiments, we could show that it is the GABA transporters that takes up the GABA too. But then I wonder, what is it doing there? So does the GABA just take up the GABA, the glial takes up the GABA and release them again? Or do they participate in this tonic GABAergic signals in some other way? So we collaborate with Dr. Justin Lee, who is has the reputation, high reputation in the glial field, and one of his graduate students had the thalamic astrocyte culture, and then put these sensing cells there, and did perform the sniffer patch, and found that this astrocytes from the thalamus, isolated from the thalamus, making the GABA without any supplement and release them. So and we could estimate the amount of the GABA from the release from the single astrocyte too. So we could see that in the physiological condition without any supplement, the thalamic astrocyte was synthesizing and release the GABA. Then what enzyme could it be? So that we look up the literatures again and found that there are two candidates, but both of them are very pathological condition. The CAT67, which is not usually only in the neurons, could go up in the Alzheimer's disease state, or the MAO-B using the putrescine as a substrate could go up, and also the Alzheimer's disease too. So we look up we did the experiment on these candidates and found that the GAD was not expressed in the thalamic astrocyte at all, and also the MAO-B inhibitors did not work at all. So while we are doing this experiment, we found that the putrescine, which is a substrate for the MAO-B, this four-step MAO-B pathway, is very high in the thalamus, even compared to the other brain area. So we begin to look into the more detailed pathway to alternate pathway for using the putrescine, and found that this diamine oxidase and ALDH, the enzyme, this two-step reaction was working to produce the GABA in the thalamic astrocytes. So to make the long story short, I'm just moving on to the molecular targeting here. But actually, from this slide to this slide, it took almost 2 years because these thalamic astrocytes were quite different from the astrocytes in other brain area. So we developed the molecular targeting method to the cell type specifically target the astrocytes, and using this physical red system, we could knock down these enzymes in astrocyte-specific manner, and we could decrease the tonic GABA level dramatically. But when we knock down these enzymes in neuronal-specific way, it didn't do anything to the tonic GABA level. So and also we applied this to the cultural system, and especially the isolated astrocyte cultures. When we knock down these two enzymes independently, it completely abolished the GABA release from the cultured astrocyte. And when we knock down the channel there, also completely gone. So we could see that the GABA in the physiological condition, the astrocytes in physiological condition, was synthesizing GABA, but it was completely dependent on the DAO and the ALDH1A1 dependent manner. And then still, we wonder if this machinery, molecular signaling pathway, is working in the adult brain too. So we use the reporting line, TD tomato, to label the astrocytes, and isolated the astrocyte from the adult mice brain, and put these sensor cells around the red astrocyte, acutely isolated astrocytes, and found they were releasing the GABA. And when we incubated these astrocytes with the aminoguanidine, which is the DAO blocker, it was completely abolished. And we could also measure and estimate the amount of the GABA released from the single adult astrocytes in the real time too. So that far, we could conclude that the thalamic astrocytes are synthesizing the GABA from the putrescine in two-step reaction and release them through the calcium activated chloride channel and act on the neurons. And we really curious that what's the function of this tonic GABA, because as I mentioned earlier, this thalamus has shows that the highest level of the tonic GABA tone in the physiological condition. But before we jumping into the functional assay, we found that realized that we do have all the tools to reduce the tonic GABA, but no tools to increase the tonic GABA. So we did a lot of tried many different methods, and while we are doing this experiment, what we have learned, when we activate the astrocytes with the different method, we could increase tonic GABA when we increase the calcium transient in these cells. And we also found that when we activate the astrocyte a certain way, then there was some average value of the tonic GABA increase by this method. And also we found that the upstream too. So the norepinephrine signaling, when the norepinephrine comes to this thalamic astrocyte, it will increase the calcium, makes the calcium transient and increase the tonic GABA. So based on this, we decided to use the tonic GABA recipient receptor specific way, and we made the calibration curve here. And we applied that based on this calibration curve, we increased the tonic GABA as much as we observed in the physiological, the enhancement. So we used this tool to increase the tonic GABA and use the other enzyme knockdown to decrease the tonic GABA. And when we apply this method to spike generation in the thalamic neurons, as you can easily imagine, we increase the tonic GABA like this, we decrease the spike generation probability because inhibition gets bigger. And when we reduce the tonic GABA level, it increases the spike generation. And then we wonder, so just if we increase or decrease the firing rate, what would it do to the sensory information processing? So we tried to mimic the sensory information processing, in the thalamus. So we looked up the researchers and found that it could make the spikes at higher than like up to 100 hertz. So we mimicked that too. Before doing that, I should explain this. So among this, while doing this experiment, we found that the tonic GABA increase in orange line here decrease the membrane resistance, so decrease the response to the upcoming excitatory input, and also make the shorter time constant, membrane response time constant here. So when the excitatory input comes, it goes back to the resting state very quickly. So combining these two, we give the high frequency input to the thalamic neurons with the manipulation of the tonic GABA level, and found that the low tonic GABA, of course, that makes many more spikes. And the higher tonic GABA decrease the spike generation. But if you take a look at the response level, spike count, in responding to the different intensity of the stimuli gets bigger and bigger. At low tonic GABA the condition, it was saturating the level of the highest level of the frequency of the spike generation. But high tonic level, although the overall spike frequency was, the spike numbers are smaller, but there was the gradual increase of the spike numbers. So we thought that this gradual increase and linear, the range is very important. So we gave if we can make the discrimination of the stimuli based on the response. So let's say we had this many of the spikes at the stimulation 1 and 3, 5 here. And at low tonic level here, the spike numbers, the histogram graphs are overlapped here. So it's hard to tell the spike, which stimuli was given to these neurons based on the low tonic condition. But at high tonic condition, the spikes histogram was completely separated. So just looking at the spike numbers, we could tell what kind of the stimuli was given. So we think that this tonic GABA level really to increase the discriminability of the spikes generation to the different level of the stimuli. So we made a simple algorithm to ask if we can really make the right answer to which stimulation is given to at the point. And if this diagonal, the matrix is brightened up, then it means that the better spike discrimination performance of these neurons. And as you can see, low tonic GABA level, this diagonal matrix is not brightened up much. But high tonic level, it was the diagonal matrix is brightened up, so the high discrimination performance of these neurons. Then another, and this is the high frequency stimulation given 10 times here. And these are the different conditions for like a low tonic conditioner and the high tonic condition. And as you can see, we express each spikes responding to each stimuli as a dot here. So many more dots here. So many more spikes are generated at low tonic level. At high tonic level, the less number of the spikes are generated. But look at how they are lined up when the 10 different iterates. So they are lined up very well here. So the spike generation is very precise when the stimulation is given. Even when the different the stimuli is given, it was made the immediate spike generation and very precise time intervals. And when we overlap all the stimuli with the spike generation like this, we found that the low tonic the condition, although the overall spike generation is increased, it was everywhere. So there was a lot of the spurious spikes were generated, but when the high tonic condition, it was always follow the given stimuli at no error, and at the very same intervals. So we think that the tonic GABA improves the temporal fidelity of the spike generation of the neurons. And we also tested that how the fidelity, temporal fidelity affects the neurons to distinguish between the two different stimuli. So we made these two independent input onto the neurons. These are just experiments to prove that these are the totally independent inputs, and made these the two independent input in different intervals, and found that the successful train rate with the different tonic GABA level, the high tonic GABA level made a better temporal resolution, which means when two inputs are coming in successive way with a very small time interval, still this neuron can tell these two independent input came or not. Okay, but low tonic level, it was hard to tell if there was the two successive stimuli was given when the intervals are smaller and smaller. So they can distinguish two independent inputs. So because this is where the most of the somatosensory information is processed, so we tested the animals with the sensory discriminability. So we knocked down the enzymes to produce the tonic GABA in the astrocyte, and found that these animals could not distinguish very similar textured sandpaper. But the very big sandpaper texture difference, still they could distinguish the big difference. And when we increase the tonic GABA with the cannular injection, with this injected the cannular, and we gave even harder task for the animals. So even the control mice could not tell very small difference in the texture, but when we injected the tonic GABA increase the molecules, they could distinguish the very small texture difference and the normal in the memory and learning and memory system. So based on this study, we could add up the new type of the cell types there. So before the thalamic the circuit, we also only studied the neurocentric way. So only the neuronal the circuit was studied, but we are adding up the astrocyte and recently even up the microglia and the oligodendrocyte to this circuit. And when through this molecular pathway, we could manipulate the level of the tonic GABA and the temporal fidelity of the thalamocortical neurons responding to the sensory stimuli, and also could manipulate the tactile discriminability of these animals. So when the low tonic GABA level, the neurons makes the low temporal fidelity spikes and low discrimination performance. When the tonic GABA level is increased, then the high temporal fidelity and high discrimination performance. So I'm looking up the time. Maybe I don't have the time to show all this movie, but probably I should just explain the main idea of this whole study. So why do we need the tonic GABA level change in responding to this sensory discrimination? When in the normal state, when we in the search mode or the detection mode, we should look around and take up the many different types of the information quickly, although this is not accurate. But when we concentrate on something and distinguish very similar information but yet different, right? That's the most important part for us to perform some cognitive functions. So we think that in search mode, probably tonic GABA level is not that high, but when we concentrate on something, we find something interesting or something very similar and need to be distinguished, and we will focus on then put the attention to that. Then attention circuit, which is the norepinephrine circuit, will be activated and activates the thalamic astrocytes and make the bigger tonic GABA level and let us to distinguish the very similar things in better way. So that's the whole story of this our study. And beyond that, as I just mentioned, we introduced the new cell types in this circuit, and also recently there was some report, the papers reporting that the thalamus is doing other than the sensory information processing. So thalamus is a huge structure in the middle of the brain, and only the sensory thalamus among the structure occupies only 40%. And 60% of the thalamus has not been studied before, and these days they are getting more attention because there was some couple of the reports, they are involved in the more higher cognitive functions such as the working memory process or social behaviors. So we think that the new data is adding up the chances for the function to study the function of the thalamus, and we keep our research on track to identify the more cognitive function, the related function of the thalamus. And as I mentioned, that this was not possible without our crew. So this is our lab members. The reason why we have so many pictures here, because since the COVID-19, we didn't have a chance to have the group photos together. So these are the current lab members. And this work was done by the talented graduate students, Hangyal, and now a postdoc in my lab. And also this collaboration with Dr. Lee and his postdoc was very successful. And the many other collaborators contribute this study and also grants. Thank you for listening.
Thanks, Professor Jung, for nice review about neuron-glia interaction in thalamocortical circuit, especially astrocytic control for GABA, tonic GABA of thalamic sensory processing. Congratulations again to get KSMCB Award for Women in Life Science. Since it is the last session today, if someone have questions, then directly ask her after this session. I'll close this section. Thank you very much.